Hello students, today I am going to explain about the tools of recombinant DNA technology. One of the parts of these tools is the restriction enzymes and biological scissors. Hello students, I am Dr. Padmavati Rao, Head Department of Botany, J.M. Patel College, Bandara. This topic is related to biotechnology, so both UG and PG students of botany can refer this topic. I'm going to explain about the tools of recombinant DNA technology in briefly. Introduction. Recombinant DNA technology is used to produce the recombinant DNAs. Generally, recombinant DNA is produced from different origins or different sources. Recombinant DNA will have the base sequences and their origin is from different organisms and are unrelated gene combinations, also called as chimeric DNAs. Chimeric DNA means it is nothing but the hybrid DNA, means its source is from two different parents. Parents means it is not a sexual reproduction through, but from two origins, different origins. Chimera combination of different sources means even in mythology, you know that in mermaids, the head is human type and the remaining entire body is looking like a fish. वो fish जैसे body रहते हैं, head है तो human type रहते हैं. That is known as mermaid. Means two combinations, means the body and the head are from different sources, human and fish. Like that, here also a new product is produced from two different sources. So such type of technology is known as recombinant DNA technology because it occurs at a DNA level. The main purpose of this is it is very much useful for the in medical sciences, agriculture, animal husbandry, and even in industry for the production of new materials, new species of plants, animals, microorganisms, like that. So first, introduction of this recombinant DNA technology. What is recombinant DNA technology? Recombinant DNA technology means recombination of DNA from different sources. We can now I am going to explain about this in a simple process. For example, here the agrobacterium, you know that agrobacterium which contains plasmids. So the plasmid is the extra nuclear material or extra chromosomal material. It has a capacity to multiply very easily, also called as plasmids. So these plasmids we can use for the transfer of desirable genes from one organism to another organism. Suppose this is the plasmid we are extracting, and this is the another one is desirable gene. Our motto is we have to transfer this desirable gene from one organism to and desirable organism or the another plant or animal or microorganism organism. So here the first, for inserting this desirable gene, we have to cut this gene or the circular DNA. How to cut it? By using some special type of enzymes. We can cut this DNA into two pieces. Then you will get a structured part. In this cutting place, we can insert this desirable gene like this. After insertion, you have to close this with the help of some gum, whatever maybe. So here also we are using some special type of biological material. After the insertion, now it has two types of DNA materials or genes. One is from plasmid and the other is desirable gene. So it is the combination of two distinct genes. So we are calling it as a recombinant DNA or the chimeric DNA. This chimeric DNA now we have to insert in the specific plant. Such process is known as recombinant DNA technology. For example, here simple thing means the transfer of a desirable gene from one organism to another organism through some biological agents is called as recombinant DNA technology process. Now, the main tools of recombinant DNA technology. Recombinant DNA technology means we are already explained, means transfer from one organism to another organism. ट्रांसफर करने के लिए फर्स्ट क्या करना है, वो डीएनए को सर्च करना, उसको कट करना, कट करने के बाद न्यू डीएनए को इंसर्ट करना, इंसर्ट करने के बाद जो काइमेरिक डीएनए को ट्रांसफर करना है। ये तीनों प्रोसेस होने के लिए 3 टाइप्स ऑफ tools we need. Those are restriction enzymes, ligases, and vectors. Restriction enzyme means biological scissors. It has a capacity to cut the DNA into small pieces. That's why you are calling it as a biological scissors. It is a biological material. It is not a non-living material. It is a biological material or the living material. And another thing, after cutting, you have to transfer it by using some special type of vehicles. Means transfer means यहाँ से वहाँ डालने के लिए some spoon like structure we need. Here the vector is the biological vehicle. Means it has a capacity to transfer these specific gene from one organism to another organism. ट्रांसफर होने के बाद वहां पे स्टिक करना पड़ेगा. स्टिक करने के लिए इट नीड्स सम glue like structure or gum like structure, that is known as ligases, biological glue. All these are the living materials. We have to extract these materials from living organisms. That's why they are biological agents. Now we are going to discuss about these restriction enzymes in a detailed way. The restriction enzymes, before going to this, I'm explaining about the history of this restriction enzymes and then classification and properties. The first one is the history. Means biological or the restriction enzymes are most important tools of recombinant DNA technology. Hundreds of recombinant DNA restriction enzymes are already discovered by different scientists. First in 1970, Arbut, Smith, and Northern, these 3 scientists, they first identified these restriction enzymes in a special type of bacteria. So the bacteria which has a capacity to protect itself against the viral infections by the presence of these restriction endonuclease enzyme. A special type of enzyme, this is the restriction endonuclease enzyme. ये enzyme bacteria में रहती है. Bacteria में रहने से a bacteria which is protected against the viral infections. Generally bacteria में viral infections होगा, कोई infection होने के बाद bacteria मर जाती, उसको resistance के लिए it has a special type of enzyme, that is restriction endonuclease enzyme. वो कैसे protect करेगी? By cutting the viral DNA into small pieces. Immediately viral DNA bacteria में attack होने के बाद ये enzyme release होगा from bacteria से. बैक्टीरिया से एंजाइम रिलीज होने के बाद वायरल डीएनए को इट कट्स इनटू स्मॉल पीसेस एंड फाइनली डिस्ट्रॉयज. सो द main property of this restriction endonuclease enzyme is only cutting of DNA. Any DNA, it has a capacity to cut into small pieces. This was first observed and discovered by these 3 scientists in 1970. So this is the base or the origin or the initiation of this recombinant DNA technology. So this is very important. Next, the classification of restriction enzymes. For restriction enzymes, several hundreds of restriction enzymes were already discovered till now, but how to differentiate these restriction enzymes? It has some specific name, that is the classification of restriction enzymes. A restriction enzymes को कैसे classify करेंगे? For example, all restriction enzymes, you know that, which are originating from bacteria or microorganisms, whatever may be. For example, HIN1, HAE3, E. coli R1. We are writing these restriction enzymes like this abbreviated form, but I am going to explain about each one like this. First, HIN1 means H. The first letter is the indication of species, Haemophilus, and the IN is the species name, Influenza. HIN1 is a restriction enzyme which is originating or extracted from this Haemophilus influenza bacterium. One means the first enzyme which is discovered from this bacterium. Like that, HAE means the first letter is Haemophilus, AE is the species name, and the third means this is the third enzyme. Already 2 enzymes were discovered from this species. This is the third enzyme. And the E. coli R1 means E. coli, Escherichia coli. This is also a bacterium. E means Escherichia and Co means coli, the species name. R, here the R is the extra part, means race. There are different race of varieties, or the race are available even in bacteria also. For a specific race, you have to use R. And the one means this is the first enzyme which was discovered first time. So this is the classification of restriction enzymes. It is very, very important topic. And then how many types of restriction enzymes are available? Generally, 3 types of restriction enzymes are available. Restriction enzyme 1, 2, 3, like that simply we can call. But the properties of these restriction enzymes are different. Restriction enzyme 1. The restriction enzyme 1 has a site-specific enzyme, but it acts on any part of the DNA and cut randomly the DNA into small pieces. So it is not useful in recombinant DNA technology. And the second type of restriction is site-specific. It has a capacity to cut the DNA at a particular site. And the third type is also a site-specific restriction enzyme, but it exactly cut on a particular site. So in such conditions, there is a chance for the destruction of some part of DNA or missing of some nitrogen bases. That's why it is also not suitable for recombinant DNA technology. So the restriction enzyme second type is very much suitable because it cuts the specific site just near the site. That's why there is no chance for the missing of any nitrogen base. That's why the second type of restriction enzyme is very much suitable for the recombinant DNA technology. The other properties, in DNA, the base sequences are read in 5' to 3' direction always. So each restriction enzyme is recognizing a specific base sequence in the double strand DNA and splits the DNA into at different sites and cut into cut at different sites and giving the fragments. And the base sequence recognized by the restriction enzyme is only 4 to 8 base pairs. Means the restriction enzyme which acts on the DNA at a particular site specific. So the length of this site specific is only up to 4 to 8 nitrogen bases long. Means it is very short fragment. Restriction enzyme cutting part site is very short fragment. So it is always palindromic type of sequence. Means we can read these nitrogen bases same in both the direction, even from left side or from right side we can read same. For example, madam means MADAM, or from this right side also MADAM, like this. This arrangement is known as palindromic type of arrangement of nitrogen bases. This sequence is very, very important. Only the restriction sites are sequence is like this, means palindromic. It must be palindromic. The range of nitrogen bases is from 4 to 8 bases only, means very short fragment. See here GGCC, and from 5' 3' side also GGCC. Here also TTTAAA or TTTAAA, same type of arrangement. That arrangement is known as palindromic. This is also most important characteristic of restriction enzyme. And the restriction enzymes DNA ligase means restriction enzyme which acts on the DNA at a particular specific site. That is known as site recognition, where the DNA cleaves. Cleave means cut into 2 pieces. So it is also 2 types. One is blunt end type and second one is sticky end type. In blunt end type, both the strands cut on the same size, like this. Here, so both the strands are in equal length and equal size. So this is blunt type. In blunt type of cutting or the cleavage, there is a chance for missing of small nitrogen bases. So it is not most suitable thing for recombinant DNA technology. And the second type is sticky ends. Means here both the strands are not cut at the same side. Means first strand cut in one side and the second strand slightly away from this side. So then after cutting, after cleavage, we will get the strands with small tail-like sticky parts. Such type of cleavage is known as the sticky ends. Sticky ends type of cleavage. It is more suitable here. I will show the diagram like this. So here in sticky end type of method, here see this here. The restriction enzyme acts on this side and here, and in the second strand here and here. After cleavage, we will get two strands like this, two fragments are like this. Means in the first fragment, second strand is like this, elongated and sticky end, and in the second piece, the first upper part is like this. So there, after reunion or the by using DNA ligase, we can stick these two by interlock method. So it is more suitable one in a DNA recombinant DNA technology, because here there is no loss of any nitrogen base. So these are the two methods of DNA ligases: blunt method and sticky method. So here the tools are three types: restriction enzymes, the most important one, and the second one ligases, acts as a glue or biological glue, and third one is vector. It has a capacity to transfer the DNA material or desirable genes from one organism to another organism. There are different types of vectors available, natural vectors. I will explain in the next topic. Thank you very much.